This protocol provides an inexpensive and adaptable system to evaluate the temporal dynamics of floral chemical cues on Colletotrichum fioriniae and other plant pathogens that infect during bloom. Novel floral rainwater collection devices enable the user to monitor natural stimulants and conditions affecting pathogen development in a near real-time, site-specific manner.
Begin this procedure with a culture of Colletotrichum fiorinae from a naturally infected host, as described in the text protocol. When colonies begin to sporulate, streak Canidia onto another V8 juice agar-containing cell culture dish to produce a high-density sporulating culture. After 7 days of growth, use a standard sterile loop to gather a small amount of Canidia from the high-density culture by lightly touching the loop to the canidial mass. Stir this into a 15 ml centrifuge tube containing 10 ml of sterile deionized water. Vortex this sample for 10 seconds. Using a standard pipette, plunge up and down numerous times to further mix the sample. Then, place a drop of the vortexed sample onto the hemocytometer and estimate spore concentrations. Adjust the concentration to 0.1 million Canidia per milliliter of sterile distilled water with a 5 ml final volume. This is referred to as the spore suspension and is only made immediately prior to use in either bioassay. To perform chloroform-based floral extraction, clean all components.